Dave Seftel, reporting from ARVO 2014 for Macular News. Would you please tell us your academic affiliations and your background?
Yes, well, I'm Dr. Dario Vasquez. I'm a medical doctor, a specialist in ophthalmology from University of Chile, South America. And now I'm working as a professor at the university, and I have my clinical practice in a public and private setting.
Tell us about the poster presentation that you brought to ARVO 2014.
Well, the poster that we are presenting this year is presenting our results from our lab that we have at the university, that we have been doing some research with retinal cells, and its specific aim was to determine the effect of an extract of a plant in the neovascular disease. Basically, we are very interested in study all the pathway that involves the development of choroidal neovascularization. So as we all know, the wet AMD is one of the worst diseases that we are attacking now as a clinical doctors. So what we did was to basically try to block the production of VEGF, which is the known molecule as the principal pathogen pathway in this disease, pathogenic. The idea was to block the VEGF before it was produced. In this case, we used the extract of the root of a plant that has been used previously in Ayurvedic and Chinese medicine, which displays anti-inflammatory and antioxidant effects. And in our lab, using human retinal cells and vascular choroidal cells, we were able to show that the production of the VEGF is inhibited by this plant. And in the same way, the choroidal vascular cells display less angiogenic activity, showing less migration and less vascular network formation.
Tell us about the compound curcumin. Where would we find it in foods that we eat today?
Well, curcumin is a yellow compound that gives the yellow color to the curry food we all known from Indian food, is now available in different preparations as turmeric on an oral preparations, right? But the problem with the oral preparations is the absorption of the drug upon oral intake is really poor.
Why is that?
Well, the problem is that the solubility of the drug is really bad. In water media, it precipitates easily. So a lot of effort has been done in the last years to develop a new formulation of this drug. Actually, if you look at some web pages, specifically clinicaltrials.gov, which is a register of all the clinical trials ongoing, you can see there are different preparation of curcumins, specifically for cancer disease, because it has been shown that it has very beneficial effects in these patients, improving the response to chemotherapics. But the new part, what we are really excited to demonstrate now, is that these effects that has been observed in cancer cells, it can be reproduced in our eye, specifically targeting this disease. So this is only an in vitro study that is giving that proof of concept. And what we are doing next is to try this same compound in a particular formulation in an in vivo model.
Now, in your in vitro experiments, what doses did you have to use to see an effect? What was the dose response curve?
We performed a dose response curve, and we observed two things. First, the type of cell is very important to know what is the response to determine the real dose. If you look at the literature, curcumin has an effect even above 2 100 micromolar on cancer cells. In our retinal cells, we demonstrated that the effect is shown between 10 and 70 micromolars. If you increase the dose, and there are a couple of publications about that, if you increase the dose in these normal non-transformed cells, there's a toxicity above 80, 75 micromolars. So the dose is really important.
So it has a relatively small or narrow therapeutic window then.
Yes, it does. Then we are really committed to develop a way to deliver this drug in a specific dose that can reach this therapeutical window.
How are you going to get this particular compound across the blood-brain barrier?
Well,
Into the eye?
yeah, well, that's a very good question. Fortunately, for us, our organ is easy to access. It's really easy to reach, because nowadays most of the drugs we are using to treat wet AMD and other diseases are used through direct injection into the eye, which is called like intravitreal injection. So that's something that allows us to deliver our drug in a more efficient way, avoiding liver clearance and other phenomena.
So tell us what the next steps would be for your research.
Well, our next step is to see if the effects we have observed in our in vitro experiments are reproducible in an in vivo model. That's some of the aims that I was pursuing in this ARVO meeting, and that is something really interesting because I already have two different groups that want to collaborate. So after we test the effect on an in vivo model, then we want to go straight forward to test its safety, basically, in human patients.
Where will these trials take place in Chile?
Sorry, when or where? Where, sorry. Mostly in Santiago and mostly in University of Chile. It's the biggest and oldest university, and we have our clinic hospital, which is a university associated hospital that has a lot of patients with this disease. And we have a big retina staff and a big retina department there.
When do you think you'll start recruiting patients?
I think the recruitment of patients won't start before 2016. I think this year and the next one, we are going to be developing the best way to deliver this drug, and we have to finish all the preclinical studies first.
Cost is always cited as an important issue in the treatment of patients, particularly in countries that do not have large resources for their healthcare budgets. Tell us a little bit about the cost implications of what you're doing.
Well, that's one of the most important and exciting points of all of this. We have seen that most of patients in developing countries are not able to pay for the treatment of this disease that we know is devastating effects. So this curcumin we are studying, it's a very inexpensive drug. And the way we are using it as a pure extract, it can be easily purchased, can be easily bought in many places. And what we want to do is to try to develop a drug that can be broadly used and that does not have the same barriers that current anti-VEGF therapy has. And well, I think that's something important since this all research has been carried out by doctors and scientists at the same university. So it's not so far pushed by a pharmaceutical company, so has been done by scientists, basically.
Excellent. Is there anything else you'd like to add?
Well, I would like to thank you for the opportunity to share some of my results. And I would like to thank University of Chile to give me the opportunity to perform this research, especially to the cell therapy, cell communications lab of Dr. Lizette Layton and Dr. Andrew Quest. And I would like to thank the the ophthalmology department of University of Chile as well.
Thank you very much indeed.
Thank you very much.